Hi, I'm Claire Highsmith doing prompt 4.2c, and I'll be analyzing the developmental network of the spines and spiky beetles. Each step in a development pathway represents the transcription pathway from gene to protein. An arrow represents a product being produced which will aid in the translation of the next one, while a T-bar represents a product that will inhibit the next step. The pathway leading to the spikes of a spiny beetle is shown above. It's important to note that two inhibitory factors cancel out each other. Think of a bouncer preventing entrance, only for a bigger, bulkier bouncer to come along and kick the first guy out of his spot to let the crowd through. If the P gene is mutated, I is not transcribed, thus K is not suppressed, which in turn will be able to inhibit E. This means this particular beetle will not have a spike at the site of the mutation. If the I gene is mutated, there will be nothing to inhibit the formation of product K, which will then be able to inhibit product E, meaning no spike will form at the affected cell or cells. If the I gene and the E gene are both mutated, it acts as a double whammy. That is, there are two factors preventing the spikes from forming. Without product E to transcribe the gene for spikes, no spikes will form. And without gene I to inhibit gene K, no spikes would have formed even if gene E had been present. So once again, we will not observe spikes at this site. If both the S gene and the K gene are mutated, spikes will be produced. Observe that the products produced by K are not needed to produce E. In fact, while S, and thus P, are needed for product I, E does not rely on the other genes in the pathway to be translated, but just on the suppression of K. Since K is suppressed, spikes are formed. Organisms would most likely benefit from having a mix of ectoderm cells, which are spine-producing and not spine-producing. The optimal spike density of spiny beetles could be induced by stress hormones released by the beetle or by epigenetic markers of its parents, leading to the methylation of DNA, degradation of RNA, or the alteration of transcription factors, altering the spikes pathway. Thank you for listening, and I hope you enjoyed.